DNA replication is an important process in determining each and every living organism's distinct features. It is an ongoing process that is constantly occurring in our bodies. The first step to DNA replication begins with the unzipping, the double helix structure of the DNA molecule. The unzipping is carried out by an enzyme known as the helicase, which breaks hydrogen bonds by that the partner bases together. These partner bases include adenine, the A, which pairs with thymine, the T, and cytosine, the C, which pairs with guanine, the G. These nitrogenous bases are also included in the nucleotide. After the unzipping, there are two strands of DNA oriented in two different directions, 3' to 5', and vice versa, 5' to 3'.
The replication and synthesis of all DNA occur in tiny openings in a DNA molecule known as the replication bubble. The strand that feeds into the replication fork is known as the parent strand, and the one that comes out is known as the daughter strand. The DNA daughter strand that is being synthesized continuously by DNA polymerase is known as the leading strand. This strand is oriented in 5' to 3' direction. This strand also faces the direction of the replication fork. In this synthesis, a short piece of RNA primer, called a primer, which is produced by an enzyme called primase, comes along and binds itself to the leading strand. This becomes the starting location for the DNA synthesis. The DNA polymerase then binds it to the leading strand and is walked along the strand, which gives it a new nucleotide bases to the new strand of DNA.
That's synthesized discontinuously is known as the lagging strand. In this synthesis, like the leading strand, RNA primers are created by something known as a primase enzyme. This is oriented into the 3 to 5 direction. Because the lagging strand needs more specific instructions to be created, it is much harder to synthesize. After this process, once the bases are all matched up with each other, an enzyme known as exonuclease strips away the primers on the bases. The openings where the primers were are then filled by additional complementary nucleotides. Then, the strand produced is proofread to ensure that there are no mistakes in the sequencing of the DNA.
Lastly, DNA ligase, an enzyme, seals up the sequence of DNA into two continuous double strands. The result of the replication is two DNA molecules composed of a new and old chain of nucleotides. Following replication, the new DNA finally winds up into a double helix shape. The new DNA produced is also known as a polynucleotide due to its composition of many other separate nucleotides. The DNA finally feeds itself into the nucleus and stores itself there. This transportation into the nucleus is assisted by nuclear pores, which are a part of a large complex of proteins and regulate the transportation of molecules between the nucleus and the cytoplasm. At last, we have the process of DNA replication in a nutshell. It is a complicated yet fascinating concept that describes a bodily function that is consistently and constantly occurring every single second. Thanks for watching.